Hello, everybody, and thank you very much for joining us on this Wednesday evening. My name's Louise Tratt, and on behalf of the British Neuroscience Association, I'd like to give you a warm welcome to this webinar, Multiphoton Imaging with Next Generation Indicators. I've been practicing that, which is hosted jointly with our annual supporters, Scientifica, and in association with Chromacity. So before we start, and whilst people are still joining, we've just got a few housekeeping rules to go over. So we'll be using the chat and Q&A function today. Do feel free to say hello and post comments and have general discussion in the chat. But if you have any specific questions for our speakers, then please make sure you use the Q&A function for these. And you should see both the chat and the Q&A function buttons at the bottom of your screen. We'll be having a Q&A session after the talks today with the chance for our panelists to answer any questions you may have. So do feel free to post your questions at any time during the talks and we'll pick them up at the end of the session. You can ask your questions anonymously if you wish to do so. There's an option to do that. Okay, and I'd like to take this opportunity to remind you that this webinar is being recorded with the permission of our speakers, and it will be available to watch on demand on the Scientifica website. And finally, just a reminder to please adhere to the B&A Code of Conduct for webinars and engage respectfully online. Okay, so without further ado, it gives me great pleasure to hand over to Dr. Christian Wilms, R&D Manager at Scientifica, who will officially kick off this webinar.
Yes, hello, thank you, Louise. So I'd also, while we wait for the last folks to join, like to welcome you to the first of what is going to be a series of webinars that we're very much looking forward to and quite excited about. And I'm particularly excited about today's topic because I'm a bit of an imaging nerd myself, to talk about multiphoton imaging with next generation indicators. A few words to introduce ourselves as the hosts. I don't want to spend too much time with that. The main focus really is supposed to be on the science here. Scientifica is a company based in England. We produce scientific instrumentation aimed at the academic neuroscience market. We pride ourselves on our support and service and working with the customers and with researchers to empower them to do the research they want to do to answer the questions they have. For this, we provide equipment, micromanipulators, stages, microscopes, and particularly multiphoton microscopes. This is why we're hosting this specific topic today, because multiphoton microscopy is one of our core areas of activity. I mentioned that our aim is to have equipment that works for the customers, for the researchers, and we endeavor to get them to do what they want to do. And the hyperscope is our flagship microscope. It's a typical multiphoton microscope. It's slimline. It has two of these scan paths in the back, and we believe it's one of the nicest systems out there if you want to have a kind of off-the-shelf but still easy to modify systems available. The reason I say that is because if you take one of these two scan paths, they're both identical. You can swap. You can use one for imaging or the other or both. They hold up to 3 mirror positions. 2 X and 1 Y mirror. One of those can be a resonance mirror, and between these there are relay lenses which image one mirror onto the next, and then that gets relayed all the way down to the objective back aperture. So if you have a new scanning technology, Bessel beam imaging, a fast Z scanning method or so that you want to employ, all you need to do is image that with a relay system onto this first mirror, and the microscope takes care of the rest of it. And as I mentioned, we have 2 of these scan paths. And the really cool thing that when I joined Scientifica particularly liked about this is that when you take one of these scan paths off, you have an industry standard 30mm cage plate that takes optomechanics from Key Optic, Newport, or Thorlabs, and you can use this to make use of the existing scan head and the beam combination and just add a scan path of your choosing. And that's especially true for fast scanning approaches. Things like facet or so can be used for this. And now if you want to do that, and this is where I come to explain why we have the second company hosting this. You obviously need good indicators that can give nice bright signals, but you also need properly powerful lasers at a reasonable cost point to be able to achieve this. And with that, I'd like to hand over to Christopher from Chromacity.
Thank you, Christian, for that introduction. Hello to everybody, wherever you are in the world. Thank you very much for attending this webinar. We feel it's quite a privilege, actually, to be invited to take part in this. My name is Dr. Christopher Leeburn. I'm the co-founder and commercial director of Chromacity, an ultra-short pulsed laser manufacturer who, for the last 8 years, has been developing a range of laser sources and optical parametric oscillators for a wide range of different applications. But where we started really was looking at developing laser sources for the life sciences imaging community, very much with the ethos of developing a light source that was compact, delivered short enough pulses and high enough average powers at high pulse repetition frequencies to become a useful tool for the life scientists that didn't necessarily need the tunability that you can get from a Ti Sapphire system. So back in 2013, we launched our 1 micron Chromacity 1040 laser system. In its original incarnation, it was a system that delivered about ½ a watt of laser light, but we now deliver a 2.5 or a 4 watt version, depending on the number of microscope systems that you want to drive. You can vary the pulse duration. We have a pair of transmission gratings that allow us to vary the pulse duration anywhere from 1 picosecond all the way down to typically 120 femtoseconds. And this has been a very successful product for us, a product that we have developed over a number of years, something that we want to be affordable, something that we certainly want to be easy to use. We have a very simple web-based user interface in order to switch the light source on and off. You can easily integrate it into your third-party APIs like MATLAB or LabVIEW using an RS-232 connection. And we've made sure that we've removed all this sort of water cooling based aspects of these light sources. And this has been a great success for us. But for the future, we also plan to bring on board a 920 nanometer fixed wavelength source. This is something that's currently under development in our R&D labs. And specifically to look at trying to be able to excite a wide range of different fluorescent markers and voltage sensitive dyes. What we've come to realize in the various interactions we've had with academics over the years is that the broad acceptance bandwidths of fluorescent markers can get you so far with a 1 micron source, but really you can do significantly more interesting science with access to a 920 nanometer source. So that's something that we are hoping to launch later this year, actually. What have we been able to do with our 1 micron source? Well, we know that fluorescent markers typically have a very broad bandwidth, and at 1 micron you can look to excite via a 2-photon, an SHG process, a wide range of red fluorescent proteins, but we've also demonstrated through interactions with the Institute of Genetics and Molecular Medicine at Edinburgh University that we can excite RFP, YFP, and also just about GFP as well, which is what you can see in this bottom left-hand series of images on the left here. So we've split out the 3 channels here. So we're able to genetic express proteins RFP, YFP, and GFP. We've also demonstrated the ability to image SHG and 2-photon. So we were looking at exciting collagen fibers that lay on top of liver samples. So here we're able to differentiate between 2 different tissue types as a consequence of using our short pulse, high average power work. But we've also done a really fascinating piece of work with cardiologists at Glasgow University, looking at comparing the low-cost fixed wavelength laser sources that we provide against a typical Ti Sapphire system. What can you achieve with this fixed wavelength source that we offer? And the kind of academics that we're working with there, we're looking at voltage sensitive dyes in very cardiac tissue, which is very opaque. So could we demonstrate a greater depth penetration by using our high-powered short pulse light source? And the answer is yes, we could. As a consequence of having greater power at the sample plane, but also being able to adjust the dispersion and maximize fluorescence at depth, we were able to demonstrate easily more than 50 and almost up to 100 microns greater depth as a consequence of using our light source. And following on from that, we've developed this sort of table here where if you don't need the broad bandwidth tunability of a TIE Sapphire, then there are a number of significant advantages to moving towards a low-cost fixed wavelength source. And I think this is really the ethos of Manuel's presentation, is looking at what we can do with these fixed wavelength sources. By all means, if you've ever got any questions, do get in touch. It would be great and fantastic to really engage with you to understand the kind of work that you're doing, to understand whether a 1 micron or a shorter wavelength laser source is in the offering in the future. And with that, I'd like to thank you very much for your time and hand over to Manuel now as the keynote speaker for this webinar. Manuel, over to you.
Hello, everybody. Thank you, Christopher, for the kind introduction. Thank you to the organizers, British Neuroscience Association, Scientifica, Chromacity for hosting the event. And of course, thank you to all the attendees for being here today. I'm very excited to see so many names continuously popping up, actually. I think we're still waiting on some people here. My name is Manuel Moore, and I'd like to use the next half hour or so to talk, as Christopher already introduced, about some new indicators and really more of a new idea and concept of taking indicators that we might already have, which are currently green and don't really excite very well with these very powerful and promising new laser sources, or old laser sources in some cases, and applying some modifications to our sensors so we can excite them more efficiently with these laser sources. So I want to start out general. And so normally when we strive to understand a system in general in science, I guess we would love to see it perform in action. And in some cases, even better if we can do that by coupling it with perturbations to understand individual parts of the system. With regards to neuroscience, and there we are. With regards to neuroscience, that means we want to see neurons as they are firing, as they are active. And an example of such a, okay, this is not, here we go. An example of such a recording is shown here. And I assume many of you here today have probably seen a similar one before. So here, genetically encoded calcium indicator, GCAMP, is expressed in neurons throughout the V1 visual cortex of a mouse, watching a movie of black and white moving bars on a screen. And sure, that might not sound very interesting to you, certainly not as interesting as the Germany versus England game yesterday, but it does elicit very reproducible patterns of neural activity, as you can see here. And the reason we can see that is that the GCAMP, this genetically encoded indicator, becomes brighter as neurons are active and their intracellular calcium concentration rises. And then as their calcium concentration goes back to baseline, they get dimmer again. So we can visualize this with a standard 2-photon microscope. But in order to get such beautiful data as shown here, we need primarily two things. We need a fluorescent biosensor, of course, as I've talked about already, and we need a microscope that can image this very well. So usually the sort of wish list for our sensors looks something like this, and I'm sure we could add more items to this list if you wanted. But the most common things that people ask for is they should be bright, they should have a high contrast, so high signal change, or often referred to as DF over F. And a little less common, but also or sometimes overlooked by people, is that you don't necessarily want to have the highest affinity, but you want to have a suitable affinity for your analyte to be within the range of biologically relevant concentration that a cell might encounter. So over on the other side, for the microscopes, we typically expect high signal to noise ratio. We want it to be fast, or by that I should say fast enough, depending on the dynamics that you're interested in imaging. If you're imaging cell division, you can probably image much slower than if you're trying to image individual action potentials or even sub-threshold voltage changes. So usually for calcium imaging, as shown here, the typical frame rate is about 30 hertz, so 30 frames per second. But as we try to image multiple fields of view, or as we try to image much faster because we have faster indicators, faster frame rates might be required. And last but not least, I would argue that microscopes should also be affordable, or as affordable as possible, to allow widespread distribution, not only to the most well-funded groups and institutes in big universities. So with regards to that last point, when we look at the price tag of a typical 2-photon microscope, we realize that the cost is largely dictated by the light source. And those are, as mentioned before, usually Ti-sapphire lasers. These lasers are wonderful pieces of engineering and can be tuned to specific excitation wavelengths that you might need for your specific experiment. But here I plotted the approximate 5-year price tag for a Ti-sapphire laser on the y-axis over their typical output power on the x-axis. And here I'm using average power. We can make different arguments also about the peak power being more related to the actual 2-photon excitation, but I want to focus on average power for now. As we already heard, there is alternative lasers, which I plot here in yellow, that are, as you can see here, much cheaper. Some of them have much, much higher laser powers, and much higher laser powers might not seem necessary to you right now, but that might enable you to either run several microscopes at the same time, or image different brain areas within the same animal, or, as we will learn later, the higher average powers, also combined with higher peak powers, have enabled new microscope modalities which are much faster, which exceed the normal standard raster scanning 2-photon by sometimes orders of magnitude in speed or in volumes that can be acquired in various ways. However, as we've also already learned from Christopher earlier, the big disadvantage of these fiber lasers, shown in yellow here, is that they cannot be tuned in their wavelength. Now, most people, most typical labs don't actually use their tunability of their lasers very much, but because they only excite typically one, maximum 2 different sensors or dyes. And usually it's a green dye. Now, if we excite only one dye and we're fixed to one wavelength, that's not technically a big problem, but it becomes a problem if we look at the wavelengths, which all of these yellow lasers here usually emit around 1030 to 1040, sometimes up to 1060 nanometers. And that is a problem if we look at the spectrum, the excitation spectrum, 2-photon excitation spectrum on the right, emission spectrum on the left, of GCamp, which is more or less representative of most, if not all, green indicators, GFP-based indicators. We see the excitation spectrum here, and you can also see I drew in a little gray dashed line at 1030, which is where some of these fiber lasers come in. Now, my colleague Kasper Podworski at Janelia and I, we set out to shift the spectrum of GCamp so we could start using fiber lasers in the future for these types of experiments like we saw earlier. And we started like probably anyone would start. We started with the best calcium indicator that we had on the market at the time, which was GCAM6. And again, also we started in a way where we started very rationally, like probably most of you would think of. We tried to graft the YFP mutations that we know from yellow fluorescent proteins onto GCAM to try to shift it yellow. And we unfortunately saw that there was only a moderate shift towards the yellow, and not as much as we had expected from looking at actual YFP spectra. So we resorted in a second step, we resorted to a non-rational screen. That means we generated a large amount of mutants, of variants of this GCAM randomly by error-prone PCR, and screened them with a method that we developed specifically for this purpose. I'm going to call it bacterial emission ratiometry. So it relies on the fact that if we express bacterial colonies on an agar plate, as we see here, if we express our indicators in bacterial colonies, as we see here, then these proteins should all excite somewhat similarly or similarly well at about 940 nanometers wide field excitation, whether they're spectrally shifted or not. Now, however, if their excitation spectrum is shifted, so should their emission spectrum. And that's something that we can detect and pick up on. Now, by eye, it might not seem very easy for you to see that there is some greener colonies here and some slightly more yellow colonies. So we used a dichroic mirror, 2 cameras, and a computer software that would translate what we see here in real time onto a larger or more broad color spectrum. And now we're starting to pick up on these spectral shifts. So now here, a red colony would correspond to a more red-shifted spectrum, meaning it ends up actually being a yellow colony. But this makes it easier for us to see, and we can pick colonies in real time under the microscope. And we can very easily, on large agar plates, we can screen thousands of colonies within reasonable times. Of course, this had to be followed up by a functional screen to make sure that the mutations that we introduced didn't mess with the sensor function. And ultimately, we ended up with a variant that had a total of 9 mutations with regards to GCAM. And at the time, we were just on time for the GCAM7 series to be ready to be released out on the market. And so I wanted to see if we can actually graft the set of 9 mutations that we found, as I described earlier, onto the GCAM7 series. And it turns out they transferred extremely well to GCAM7S and GCAM7B, giving us a slightly slower and a slightly faster yellow variant of JGCAM7, which I'm going to refer to as JYCAM for yellow 1 and JYCAM1S for the slow version of this indicator. And here is shown the actual spectrum of both of these variants. They have virtually the same spectrum, so I only plotted one. The yellow shifted spectrum of JYCAM is the yellow-orangey line, and you can hopefully appreciate that now we can efficiently excite we can presumably efficiently excite these at fiber laser wavelengths above 1,000 nanometers. And I don't want to bore you with all the characterization in neurons, and I want to jump right into in vivo data. I understand a lot of you are neuroscientists and love to see in vivo data. So here I'm going to show a relatively standard prep where we inject AAVs encoding for JYCAM and JGCAM. So here it's JGCAM7S and JYCAM1S. in different animals, and we compare them side by side under identical imaging conditions at 1030 excitation. So here we can see a respective tuning map during visual stimulation using, again, this standard stimulus of white and black moving bars. And on the left, we can see the performance of JG-Camp 7S, and on the right, we can see the yellow version JY-Camp 1S. And I hope from these images you can already see that we get a lot more robust directional tuning. And when we quantify this, as seen here on the right, we achieved almost 3 times the signal to noise ratio, about 2.5 times the signal amplitude, or DF over F, and more than 2 times the brightness, the baseline brightness of the indicator. And we also performed similar comparison in the brain of flies. We made a GAL4, so we combined a GAL4 driver line called MY1 with a JY-Camp1 reporter line, and imaged them while they were given a full field visual stimulation of basically just light turning on and off in their field of view. Prep looks something like this while we image the fly, where I should mention the cuticle is removed for easier access. Now, the typical columns in layer 8 to 10 of the medulla of the fly that would typically get labeled with this MI1 driver line, reporter line, is shown here. And while we don't, in this prep, we don't see a big difference in baseline brightness, we do see the same trend for DF over F, which is almost 2 times as large. And I have representative functional traces plotted up here on the top. And so we were very excited about the performance of this yellow shifted version of G-Camp, or this yellow calcium indicator, in vivo. And I would argue the best thing is still to come, though, where we realized, and Christopher hinted at this earlier, that you can co-image yellow and green fluorescent proteins at the same time at this excitation wavelength. So we thought, looking at the spectra, and I show them here again, we might be able to co-excite a yellow and a red indicator using a single laser light source. And so, as I said here, I plotted the left graph is the excitation spectrum again, where we see JY-Camp and the best-in-class red calcium indicator JR-Gecko 1A. Both excite very well at this 1030 wavelength that fiber lasers produce. And on the emission side of the spectrum on the right, you should be able to appreciate that their emission peaks are well enough separated for us to actually separate them using a dichroic and know which of the indicator gave us the signal in the end. So we devised an experiment that several people have attempted before with very varying success, I should say. And it's shown right here. My former colleague, Jiaji Yang, helped me to inject an axon-targeted version of JY-Camp into the DLGN, or dorsal lateral geniculate nucleus, which has direct projections, direct axonal projections up to visual cortex. And in visual cortex, we express the red indicator JR-Gecko 1A via a transgenic Thy1 mouse line. Now we can image in visual cortex and observe pre and post-synaptic calcium transients simultaneously. And this is a representative video what this looks like when we again present our favorite stimulus of black and white moving bars to the animal at different angles. So when we look at this, ideally, with all this activity, we would like to be able to find the few sites where the presynaptic signal and the postsynaptic signal are correlated, which would then suggest that those are sites where putative synaptic transmission is actually taking place from the cortical ascending neurons from the DLGN over to the neurons in visual cortex. And as a matter of fact, when we run this very same experiment with JGCAM7S, which at the time was the best indicator in green, and run a pixel-wise cross-correlation analysis between the green channel and the red channel, it does not actually, we find no significant difference from a random shuffled model. So in other words, what that means is we were basically, in this setting, unable to find these site of synaptic transmission. Or yet in other words, we pretty much failed to find the needle in the haystack. Now with JYCAMP, however, we can run the same experiment, we can run the same analysis, and automatically filter out pixels where coupled activity in overlapping components of the yellow and red channels is significantly more cross-correlated than random chance. And you can see a zoom in of such a hotspot of cross-correlation here, where we pulled out what I would argue is the needle in the haystack. We find these synaptic sites where we see pre and post-synaptic activity being clearly correlated. With this very, very exciting implications for traditional raster 2-photon microscopy, I would like to come back to this slide that we started with in the beginning and discuss a little bit more about the need for microscopes that can image the same field of view, but at much faster speeds. For example, with regards to voltage indicators. And also there's plenty of ongoing efforts to push not the speed on the same size of field of view, but to expand the field of view out and image much, much larger volumes in one animal at the same time. And I present here an arguably very small subset of the growing number of exciting new microscope modalities that have pushed these boundaries over the last years. One feature they all have in common, and I'm happy to say this specifically during today's talk, is not a single one of them uses a Thai sapphire laser, but they all, without exception, use fiber lasers as their light source. So traditional 2-photon microscopes can scan a single point across the field of view in this raster type fashion. And if we look at this first example on the left here called SLAP microscopy, or scanned lined angular projection microscopy, developed by the Podgorski lab at Janelia, here it's not a single point, but it's a line of excitation. It's an excitation line focus that is swept across the field of view at different angles. And then computationally, the fluorescence from every pixel is calculated back. And that allows us frame rates of about 1 kilohertz for a similar field of view as you would know from your raster scanning 2-photon. Now, the second example here is called FACET, and Christian already mentioned it earlier in his short intro. This technique was first developed by Kevin Xia from University of Hong Kong, and for 1-photon excitation, and then adapted for 2-photon excitation in the lab of Naji at Berkeley. And FACET relies on a set of almost parallel mirrors that basically split an incoming laser pulse into a series of subpulses that then enter the objective at slightly different angles. And so on the imaging plane, what that means is we're going to have a line of spatiotemporally separated focal spots. And this line can now be scanned across the field of view, which practically reduces the frame rate down to the line. scan rate of a typical 2-photon microscope, and it's achieved a staggering frame rate of up to 3 kilohertz imaging speeds. And the third example I want to highlight today is Mark Schnitzer's lab from Stanford University came up with a microscope that basically has 400 individual foci that it excites at the same time. There's no temporal separation of these foci, but only a spatial separation. So they use a camera to image these and scan the focal spots over small distances. And the very last example I'm going to highlight is actually still on preprint and is not yet published as a peer-reviewed paper, but it's a very exciting new microscope called light beads microscopy from Ali Pasha Waziri's lab at Rockefeller, where the way they achieve it is very different. But if we go back to facet, it's actually kind of similar in the overall concept, where we have a line of slightly temporally offset foci that can then be scanned. Now the foci are now, the line of foci is not along the x-axis or y-axis, but is now in the z direction. So this allows Ali Pasha's lab to perform video rate volumetric imaging of large areas of cortex, and together with reducing the spatial resolution to individual somata instead of subdiffraction limit or diffraction limited 2-photon foci, this achieves a staggering simultaneous imaging of 1 million neurons in cortex within a single imaging session. Now, as I mentioned, all of these microscopes rely on fiber lasers as their primary light source, most of them for their high energy pulses, which we haven't talked much about, and I hope that we will be able to go into that a bit more during the discussion. But to overcome the problem of suboptimal excitation of green sensors at fiber laser wavelengths, some of these groups have come up with sort of an engineering approach to the same problem. They use an optical parametric amplifier, or OPA, to change their wavelength to 920, which is, as we've discussed, of course, much more suitable for green indicators. Now, while this is a great idea, OPAs are even more expensive than your typical Ti sapphire laser and are even more in need of regular maintenance and alignment. So, and then last but not least, they have tremendous power losses throughout. So if you look at the example of facet right here, we start out with a 35 watts fiber laser, and after the OPA, we end up with 2 watts post OPA. And combined then with the high number of optical surfaces in the actual microscope, this often means that these microscopes are operating close to their respective power limits. And I would argue that with yellow shifted sensors, these microscopes can use the output of their fiber lasers directly, not losing any power throughout the OPA. And at least with minimal modifications, maybe pulse compression, we would open the field for easier distribution of these microscope and commercialization potentially, as Christian hinted at, and even better and potentially even greater microscopes down the line if we can work with the fiber laser directly. Now, just in the last few minutes or so, I want to tell you I'm very, very excited to tell you that JYCamp1 has made it in the hands of some of these labs, and there is actually a first preprint available as of a few weeks ago from Jerry Chen's lab at Boston University, and in there, what they call quadroscope, they simultaneously scan 4 fields of view across large cortical areas using a 1040 fiber laser, and they excite JYCamp1 with it and detect almost 600 neurons across different fields of view, ranging from M1 motor cortex to S1 and S2 somatosensory cortices. During I believe it was a whisker-based texture discrimination task they were applying as a stimulus here. And I'm not going to go into the details of all the very exciting findings that they made with this, but I would really like to point out that this type of data was made possible by the combination of amazing new microscopes that use fiber laser wavelengths together with new indicators. And very, very last thing I'm going to talk about now is where does that leave us with regards to the field of sensor development? Now, the discovery of GFP was accompanied with the development of affordable light sources that excite at 488 nanometers. And that has, as probably most, if not all of you know, enabled the broad spread of fluorescent microscopes anywhere. In the case of 2-photon microscopes, we have now these powerful light sources available at low cost if we only had yellow indicators. So the situation is kind of the other way around. And while I have clearly shown one of the examples where the yellow shift was admittedly not straightforward, and we had to resort to non-rational methods, it doesn't have to be that complicated. And to show you that, or to really make that point clear, I want to show you the examples of a yellow eye glue sniffer and a yellow eye acetylcholine sniffer, both developed in Lauren Luger's lab at Janelia, where the yellow shift was extremely straightforward. So here we really rationally, or I shouldn't say we, my friend Johnny Marvin, grafted known YFP mutations onto a green indicator, and it worked pretty much almost out of the box. So I propose that for a lot of indicators, we might be able to make this yellow shift with very, very easy steps that most labs with a little bit of molecular biology experience can perform. And sort of on the very right, I want to just also hear your opinion in the Q&A session, which we're going to start very soon. What is your favorite indicator? What would you like to be able to image? And do you think would be a great indicator to be able to image at fiber laser wavelengths? Clearly, there is always a demand for more neurotransmitters. There's also a lot more neuropeptide sensors coming up. And of course, the always omnipresent question is always, how about voltage indicators? For 2P imaging of voltage, I'm going to say it's always a little bit difficult because a big class of voltage indicators, which are opsin-based indicators, seem to turn off when illuminated with these long wavelength lights that we get in 2P excitation. But I also hear that there is new versions of the non-opsin-based voltage indicator ASAP, and they are already slightly yellow shifted. Whether that's enough, we'll have to see, but I also know from Michael Lin that there is ongoing efforts to optimize these sensors further for fiber laser excitation. So with that, I would really try to keep my acknowledgements somewhat quick to leave enough time for questions that you have and for the exciting Q&A I'm looking forward to. I want to mention specifically in my acknowledgements, Kasper Podgorski and Eric Schreider, my supervisors and bosses at Janelia, the Genie project, under the leadership of Doug Kim and Alan Wong, but there's also a number of undergrad students, which I'm excited to name here, that have contributed significantly to this, as well as collaborators, administrative staff, and the core facilities at Janelia. So thank you again to the organizers and to you for attending, and please do stick around for an exciting discussion session. Thank you. And I hope we're going to get Christian back online. Hello, Christian.
Hi, Manuel. Fantastic, really, really interesting and fascinating talk. Especially nice to see a technology that was released only in physiological terms recently already being put to use and leading to upcoming publications. We have a set of questions coming through. Should we start with those, or?
Yeah, let's start with those.
So Ken Berkland has asked, well, thanks you also for the great presentation, and is asking if you've compared JY Camp with Haruhiko Bito's X Camp Y.
Yeah, so that's a great question. We have not done back-to-back comparison, or I haven't. I have heard people trying or experimenting with both of them. I don't have data that I can show right now where I can where they would compare back to back. The X Camp Y, I was going to say XG Camp Y, just a matter of habit. X Camp Y might be a similarly good option for, or it might be a similarly good, well-suited backbone for this shift towards yellow. I don't feel entitled to comment on which one is better, if that's what you're looking for, though, unfortunately. I think this is a very good question for Christian, actually, that came up here from Nikolaus Droseros, and I'm hoping I'm pronouncing that name correctly. What are the biggest challenges when connecting a 1040, presumably fiber laser, to a microscope?
It's actually not that challenging. It's very, very straightforward. We've done this repeatedly in several workshops and courses, and we've specifically also used the Chromasity 1040 for that. And there's really no difference to the use of a titanium sapphire, except for the fact that it takes up less space on the table. It's very straightforward. Free space couple. You send it through a pocket cell or an AOM as an attenuator, through a beam expander over a set of mirrors into your periscope and of the microscope, and you're done.
It's got to the point now that we don't need to send an engineer to Scientifica to do this kind of work. They can easily do it themselves.
Yeah, I think the last 2 or 3 times we just dropped, one of our colleagues just dropped it on the table. It's really, they're wonderfully straightforward.
Placed on the table.
Placed, yes, no, not dropped. They're not literally dropped.
Yeah, I know.
Yeah. So, and I can actually comment on that myself. We were lucky enough to have a Scientifica system for demo at Stanford University at some point, and had the fiber laser come in, and I was there while the fiber laser was being installed on the system, and it was sensationally simple. So the difference here, particularly for me, was I've seen fiber lasers get installed, and I've seen Ti-Sapphire lasers get installed. And it begins with the fact that you can, no problem, lift a fiber laser by yourself as a single person. And I'm not talking, you don't have to be a bodybuilder to do that. You can lift it out of the box, put it on the table, and get started. And it is really that simple. With a Ti-Sapphire laser, if you get it delivered, you're going to have to set up the entire liquid cooling system, and you basically need an expert to do it, I would argue.
We've got a number of customers who like to share the laser across different labs. And so the system can be picked up and taken across from different rooms from time to time. And there's been no issue there. So I understand your point. I see another question's coming from Juan Carlos Boffe. Have you tried simultaneous excitation of JY Camp with JR Gecko 1A, also with a single 1030, 1040 nanometer laser source? And would that work for two color imaging?
Yes, absolutely. And so that's actually what the data was from that I showed earlier. So this experiment where we did axon targeted Y-Camp and somatic or non-localized somatic filling JR Gecko, that in visual cortex, that was data from exactly this type of experiment. So we excited at 1030 nanometer wavelengths, and imaged both colors and were able to separate them with minimal crosstalk in the two channels. Yes. So that's exactly what I think is a great application of these yellow indicators, exceeding just the enabling fiber laser wavelength imaging. Yeah.
Yeah. So one thing that I was wondering about as you were talking about, especially the how you came to the design of JYCamp, but also then how it was adapted to the similar approaches were adapted with other indicators like glue sniffer, white glue sniffer, and the acetylcholine detector indicator, is what the future is. I mean, if we look at GCamp, it's gone from GCamp 3 to 5 to 6 to 7 to now 8 in a remarkable amount of time. So do you see, and obviously every time their performance increases, and it's clear that it would be really good to see this with things like YCamp as well. Do you see it as a matter of you take the next generation GCamp and introduce those 9 mutations and adapt, or do you see it as a completely independent development moving forward? Because obviously that has implications, because one can be done by small labs and the other needs the facility to actually do the level of screening that's required.
Right. So I think that's an excellent question, and one that we have very extensively discussed internally too, right? Especially with now GCamp 8 having been released, or not yet published, but released on all platforms, @Gene, etc. It's a very obvious question. So can we make a yellow GCamp 8 by just popping over those mutations? And I haven't done it. I actually would have to, and I've been meaning to call up Lauren and ask him if this is something that they're doing currently. And I haven't gotten to do it, haven't gotten to call him yet, but I would envision that so the answer is probably it will work. So, and the reason I say that is we initially screened for these 9 mutations in a GCamp 6 background. And we took those 9 mutations, and without any further adjustment, we morphed them over onto GCamp 7 and saw it work like a charm out of the box, at least for some of them. So we did not see this transferability work out really well for GCamp 7C, for example. So 7C, for those of you not intricately familiar with it, it's the variant that has an extremely large delta F over F as a result of its extremely dim baseline brightness. So it seems that that particular backbone was not as amenable to these modifications, to these yellow shifting modifications. We actually created a working indicator, a working calcium indicator, but we did lose this very, very low baseline fluorescent brightness feature of it along the way. So that's something, there might be cases where the direct transferability is not as straightforward. And I think GCamp is probably one of the prime examples of where it might be difficult. But I foresee that a lot of other indicators, especially seeing how extremely easy it was to shift the yellow, the sniffer category of indicators over to the yellow. I'm very motivated and very confident that a lot of sensors will be very easily adopted to yellow. And I'm also very happy that I saw several very prominent tool developers in the audience today, and I hope they will take that little hint and for their next sensors consider making a yellow version and publishing it alongside, just to make that threshold even a little lower for neuroscience labs that might not have the capability to do excessive molecular cloning to adopt these techniques even faster. Yeah.
I think as I can see 2 more questions. We've got, I think, 5 minutes before we get, well, Zoom decides that we're finished.
Yep.
So one is from Dirk Luchtmann, who asks if that we talked about JYCamp in 2P, if you could see it being used in 3 photons, experiments as well. And I'm guessing the idea here is that it gives you the advantage of, rather than being able to do 2-color imaging with a single excitation line potentially, rather than right now, if you want to do 2 colors, you'd have to basically cook the brain.
Yeah, that's a wonderful question, Dirk. Thank you. So in order to answer it, I have to kind of tell a little bit the background. So with 3-photon excitation, for green we use typically 1300 nanometers, and for red dyes we use 700 nanometers. Now you can already see by these largely different numbers, 1300 versus 1700, there's a gap of 400 nanometers. It's something that is very unlikely to work with a single excitation wavelength, even if you consider coming in right in the middle, say, right, with a yellow indicator. You would probably place that one at, and I don't have the exact 3-photon spectrum here, but we would probably place it at about 1500-ish nanometers. And so there's two drawbacks with that. One is, A, I don't think it would allow you co-excitation, and B, there's actually, so if you look at the 3-photon transmission spectrums through tissue, you actually see that the penetration depth is sort of has a peak around 1300 and has another peak at 1700, and actually has a dip, a little valley of less optimal penetration through tissue in between. And I think, if I remember correctly, that has to do with the absorption of hemoglobin at that wavelength. So in a sense, JY-camp is probably not going to do better than JG-camp, and it probably wouldn't allow for dual color imaging. Now, that being said, there's a new paper that just came out by Chris Shu's lab, where they show that you can actually excite at 1300 nanometers. You can co-excite green and red indicators already very efficiently. And that has to do with a larger energy transition that is being excited in the red fluorophores. So traditionally, we would think 1300 for green, 1700 for red, but it turns out 1300 works for both green and red very efficiently. So if you're looking for 2-color 3-photon imaging, that's probably the way to go. Yeah.
I'm conscious of the time. I don't want to be in a situation where we're kind of cut off mid-question. So Louisa kindly posted our email addresses. We're really happy to get back. I'm aware of the 2 questions that have been posed that we haven't gotten to yet. We will be getting back to you on those. And if you have any others, please feel free to contact us. Thank you again, Manuel, for really fantastic talk, very interesting discussions, really nice questions to go with that as well. And thanks, Christopher and Chromacity, for supporting this. And definitely also thanks to all of you for sitting through and driving this discussion.
Thank you, Christian. Thanks to everybody that was here today. Thank you very much.
Thanks.
Thank you.